All right, now we're recording. Here we go for posterity. Welcome everyone to our Division of Biological Sciences seminar. We're here at the University of Missouri. Today we're here for the public seminar for Sean Abrams as part of their requirement for their PhD defense. I am Chris Pierce, pronouns he, him, and I am Sean's graduate advisor. It would be great today if we were all in a big, large lecture hall in person so we could all see each other. However, today we come to you virtually. So we are thanking all of you for being here from across the US and indeed across many countries. And I'm especially grateful for all the friends and family who are here today to see Sean's seminar. Sean Abrams is from Sunrise by Fort Lauderdale in southern Florida. Sean often credits Pam Krause, a high school teacher trained in plant biology, for encouraging Sean's inner plant nerd and pointing them to an Everglades Restoration and Environmental Science magnet program. Sean then attended the University of Florida and worked with several researchers there on a number of topics. Sean started as a researcher as a freshman, and so was able to go through a few different labs during their time there. I first met Sean at a Botanical Society of America meeting in New Orleans in the summer of 2013, and again the following year in Boise, Idaho. And in both cases, I saw Sean, even as an undergraduate, already a very poised and confident speaker for both research, helping and leading workshops with Dr. Pam Soltis. Sean arrived here at MU in the fall of 2015. The whole cohort of 2015 has had unusual circumstances of being PhD students, and so they all get extra points for the last 5 years of being here. Sean's goal was learning phylogenomics, comparative genomics, and metabolomics, and they have been a real leader in interdisciplinary research in our lab, from growing plants to organizing databases to really developing their own omics pipeline that they're going to talk about today. Sean has been a phenomenally strategic thinker with a long-term plan and a very successful grant writer. I'm just going to tell you about two of them. Sean won a National Science Foundation Graduate Research Fellowship award to support their PhD. But on top of that, they got a really nice supplemental award to go to Wageningen University in the Netherlands to work with Dr. Eric Schrans and collaborators for a year. And during that time, Sean learned a cutting-edge phylogenomic Cincinnati program that Sean will tell you about today. This is such a sought after skill that everybody now wants Sean as a co-author on their papers. And for example, one of them came out yesterday, led by Drs. Ella Katz and Dan Klebenstein at UC Davis in the journal eLife. Sean's also was the leader of a grant from the National Geographic, which was to go to the Mediterranean and collect all the plants. And I imagine Sean being on a nice sunny boat or a sunny island, but then COVID. So hopefully next year, Sean will be able to resume their field work. To date, Sean has 3 manuscripts out. I think they'll describe one today and various others in publication. Teaching, mentoring, and service. So without a doubt, if it was my ability, I would give Sean another PhD for this role. Sean has been very, very thoughtful about sharing their science. You can just Google Sean's name. Sean's already famous for being on podcasts, talking biotech, organizing local things, and just go check out Sean's own YouTube channel. Sean's track record in service really goes back to when they were high school students as part of the Preparing Leaders and Nurturing Tomorrow's Scientists or Plants program for botanists from underrepresented groups. One major outreach that I knew Sean from early on was from their work with the UF Gators Reaching Out with Botany or Grow Botany program while they were undergrad, and they also visited elementary school classrooms. And upon arriving here, Sean was also involved in a national effort with outreach out in STEM or OSTEM, both as a president and as a vice president. So Sean already has a national, if not international, presence and service. And then we come to this last year. There are no words. that can describe all the things that Sean has done for everybody this last year, whether it's our weekly lab meeting, our weekly journal club, Biosci Divisional Council, people in our dean's office. Everybody has reached out to Sean, and they have done a phenomenal amount of work for everybody. Sean has had a direct impact on the hearts and minds of many people for just being themselves. So Sean is a passionate advocate for inclusion, diversity, equity, and if you want to see for yourself an example of Sean's work, I invite you to go look at Sean's YouTube video from last week on the Black Botanical Legacy. And I want to give a shout out to our division director, David Scholes, for providing research support to let that happen. The next steps is Sean is going to move on to a postdoc position at Yale University, where they'll be working with Dr. Erica Edwards in collaboration with Dr. Jen Weiscaber at Purdue University. And lastly, before I throw the mic to Sean, our whole lab will miss Sean, past, present, and future. Everybody leaves their footprint. And we'll also miss, I don't know if we'll see her today, Sean's cat Jaja, because all the extended land family and the dogs and cats of the plant world are part of our group. At the end of the presentation today, please, and I'll remind you at the end, put your questions, not in the chat, but in the Q&A section of the talk. So with that, please give your full attention to Sean Abrams. So Sean, take it away.
Thank you so much, Chris. I'm glad I held it together and didn't cry. For this talk, as Chris said, when I came to graduate school, I was really interested in phylogenomics and genomics, and I think I really got to accomplish that goal of expanding my skill set to include those. This will involve some more complicated topics up front, so I'll hopefully break those down with some analogies throughout to make it a little bit easier for everyone, but I'll go ahead and get started. So the group of plants that I work in is the order Brassicales, and you'll know a lot of these plants as members of major crop systems. So in the Brassicaceae, the mustard family. Oh, I know I can change my pointer here. Yep. So you have of course, your dogs of the plant world, Brassica oleracea, but a lot of different crop species eaten across the world. In the Cleomaceae, there are super crops, and then in the Caperaceae, you also see capers, and so a vast diversity in this family. But part of the diversity that we see in the family is associated with their complex genomic history. They have what you call whole genome duplication events or polyploidy events. In this case, event beta and event alpha. Beta is shared by the entire tribe except for Carica, and then alpha is only shared by the Brassicaceae. So what is whole genome duplication? So in a normal diploid, maybe normal is the wrong word, you expect to have two sets of your chromosomes, but if an error in your meiosis occurs, there can be unreduced gametes that then lead to the offspring having twice as many chromosomes as the parent did. This is generally a very intense process for any organism, but if you survive, you have access to a lot more genes to adapt with. And on top of that, if it was a single species, you would have an autopolyploid. You can also do this in a way that is a hybrid. So now you're having not only twice as many genes, but genes with different histories and potentially different functions coming together in a novel context. This phenomenon is associated with a lot of key innovations from the vertebrate spine to the angiosperm flower, and hopefully I'll tell you a bit about some other innovations that it's been a part of today. What allows for this innovation over time is the normal process that occurs following a gene duplication of any kind. You either get degeneration and loss, so you wouldn't actually see anything happening there because you would just lose the extra copy of that gene. You would get subfunctionalization. So potentially those two, that extra copy, they split whatever job they were doing before. So now each gene is working less as hard. And then of course, neofunctionalization, where you get a completely new trait that comes from an adapted gene. Gene duplication can also, when working with phylogenies or species trees, right, I'm trying to understand species relationships, and species trees are made up of smaller sub-gene trees that in total give us what we would consider to be the full species tree. But because of gene duplication, it can get a bit more complicated than we would prefer. So in this case, so here in black, we have a gene tree that follows this species topology. But if we were to, say, have a duplication event, and throughout the talk, I'll use these symbols to refer to whole genome duplication events, so you can look out for that. In this case, it gets a lot more complicated. You probably can't even follow this figure anymore. But if you see, like, you have gene loss events from those duplicates, right? Some of those duplicates disappear. So when we're just using sequence comparison alone, it can be very difficult to pick out which genes are the paralogs, the duplicates, and which genes are telling us what the species tree is, right? And so a way of dealing with this issue, well, is writing a lot of papers about it. But a way of dealing with this issue is informing your analysis using synteny. So synteny is the physical order of genes in the genome. It's which chromosome it's the address of the chromosome for each of your genes. So this is actually my 23andMe analysis, but you can see, like, if I had a gene on my X chromosome, that would be its address, X chromosome in this location. So if I had the first analogy, so if I had a piece of mail that I wanted to send to someone, say this is me using sequence similarity approaches, I would say, okay, can you hand this to Black Panther for me? And so you would run around town looking for Black Panther, and maybe you find T'Challa, or maybe you you wind up in a zoo someplace and you hand it to that Black Panther. And so in that way, it can be a bit confusing just by appearances. What is the true, what genes are telling you what in terms of ancestry. Adding in synteny gives a house, right, for our analysis, allowing us to associate a specific location. In this case, T'Challa lives in the blue house, so you can go to the blue house, and then you know exactly what you're talking about and what the importance of delivering that letter would be. Okay. All right, so as a broad overview for my talk, I'll discuss the role of gene duplication in the origin and diversification of MAM. So I included the Caperuaceae here. I'm not actually going to talk about the Caperuaceae. I'm mostly going to, for the insights into the Cleomaceae in terms of MAM, and then talk about the Brassicaceae and the dynamics for the MAM IPMS gene family there. And then finally, I'll end with the smallest phylogenetic clade, the tribe Brassicaceae, and how utilizing phylotranscriptomics has helped with some insights there. Okay, so as Chris mentioned at the beginning, I got to travel to the Netherlands for 7 months, work in the lab of Eric Schrans, and learn some really great genomic techniques. This was funded by both the NSF and the NWO, and as a part of my GRFP fellowship. So one of the key aspects of plants in this family are their taste, right? So that taste compound that we like and sometimes don't like, that wasabi flavor, that yellow mustard, is what we'd call glucosinolates, or mustard oils. The this is not only just found as condiments, but it makes up the flavor profile of a lot of different major crop species, cabbage, arugula, radishes, and it is also really important for the interactions that these plants have with their natural world. So in this case, you might have a butterfly that wants to lay some eggs on these crop species, these insects. They're going to munch, 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 and depending on whether or not they've adapted, they're generalists or specialists, the defense compounds will try and will aid the plant in fending off this attack. When I came into the lab in 2015, this paper had just come out by Pat Edger that looked at how gene duplication, and particularly these polyploidy events, have impacted the diversification of glucosinolates, but they largely focused on the broad aspects. And I was really interested what was going on in, so if you see here, this is where the bulk of the species of the order are. And so I was like, okay, well, what's actually controlling a lot of these novel structural elaborations? What is the big shift that's happening here, specifically outside of the shifts that we see in substrate? So the glucosinolate biosynthesis, and I know not everybody is super good with biochemistry, so I will limit it as much as I can, or simplify it as well as I can. You can break it up into three major steps: a chain elongation step, the formation of the core structure, and the novel structural elaborations at the end. That Edger paper focused largely on the formation of the core structure and changes there. For my analysis, I wanted to look at this chain elongation step. This is I often use a car analogy. So if you're building a car, the formation of the core glucosinolate structure is you're actually assembling the vehicle. For the chain elongation, that's how those are the materials that go into the process. So MAM is changing the metal that will eventually become the engine or the door or the tires, the rubber for the tires, right? We're making more advanced materials that will then go into the creation of the defense compound. So like I said, so this is a methionine amino acid, which is one of the inputs for this pathway. And what MAM does is we'll take this single carbon bond and it will add a carbon, add a carbon, add a carbon, add a carbon. And so you can get these more elaborate glucosinolate structures with added carbons, kind of like a Swiss Army knife, right? The more sections in it you have, the more tools you can add onto your Swiss Army knife. Most of this work has been done in glucosinolates in general, has been done in the Brassicaceae, and largely between Arabidopsis and Brassica oleracea. So that's your cabbage, cauliflower, and Arabidopsis is a model species. The main phenotypic difference between these are that Brassica, it can produce 3C or 4C glucosinolates, but it can also produce 3C and 4C glucosinolates at the same time, where Arabidopsis does not have this and function. But they can go up to very high carbon amounts for their prep. MAM is a specialized metabolic gene that has come from, so it initially was a duplicate of the primary metabolic gene IPMS. This is important because we actually didn't know how IPMS transitioned into a MAM type function, considering they have very different constraints. IPMS is involved in leucine biosynthesis, while MAM is doing this specialized defense compound creation. So part of what I wanted to do is to suss out how this transition actually occurred between these two groups. And so I've mentioned whole genome duplication. It's a full context copy where you get a completely doubled of your genome. The best analogy came out of a class that I was taking, that Chris and Gavin Conant were running. I believe that was that class, and it was, who was it? It was Jessica Neal. Jessica Neal came up with this. So this metaphor is, think of genes like cake. So you're making a cake. If you want to double the recipe, that's your whole genome duplication, you just get more cake, which is good. Maybe you want just a bigger cake. Maybe you want to make 2 cakes out of your recipe. But certain genes and certain ingredients don't like being duplicated in specific ways. So there are smaller duplications, like near duplications or tandem duplications. These duplications are like if you wanted to add more eggs or add more sugar to your recipe. You get a sweeter cake, you get a more dense cake, you can get moist cake, right? There's a lot of different cakes that are still edible, but you wouldn't want to add salt to your cake, right? You just double the salt amount, then you get salty cake, and that's not very good, and you have to throw the cake out. Or like a mistake I made very recently baking, adding too much baking soda. You don't want to double baking soda without doubling everything else. And so when you're talking about these singular duplications, so these near tandem, and then a transposition duplication, those have that kind of effect. Although a transposition gives a novel context as well. Okay, oh, and then this is the everything is cake meme. And this happened last summer. So just think about everything is cake, all genes are cake, and then potentially brassicas are cake too. So the goals of this analysis were to characterize the role different gene duplication types have played in the expansion of this gene family and the origin of man, and then place these in the understandings in the context of MAM diversity. So this method that I went to the Netherlands to learn about is a phylogenomic synteny network analysis, where you on one end you're generating a synteny network of genes. So this is, you're comparing genomes so you can get the shared addresses between all your different genomes that you're using. And then you're building a phylogeny using that sequence similarity that I talked about in the beginning, the A, C's, T's, and G's, and building your tree, and then you're overlaying your synteny information for whichever specific gene family you're interested in on top of that. I used 35 genomes from the order Brassicales to do this. This was the first study of its kind that utilized the Cleomacea in this way as an outgroup for understanding glucosinolate evolution. And so in the analysis, so you have to select specific domain targets when you're looking for gene families. And so domains are conserved regions of the gene that offer that specific function. So in this case, the HMGL-like pyruvate carboxylase domain is what is shared by both MAM and IPMS, and adds that adding extra carbon part, okay? But IPMS has an extra little bit at the end, this leucine allosteric dimerization domain. And what this does is it allows IPMS to link up to other enzymes, similar enzymes, and form a homodimer, which is really important because, as we'll talk about later, these are going to want to pair up with each other as they, in order to do their biological function, where that is not necessarily the case with the MAM enzyme. So in my full analysis, I did identify 3 clusters. So we expected an IPMS cluster and a MAM ancestral cluster, but I also identified a novel, what we're calling a transposed MAM cluster that is lineage specific in the Brassicaceae. So a novel location for these genes to be acting and potentially changing some of their functions and interactions. I want to highlight here in the Cleomacea. So I mentioned this is the first study to include the Cleomacea in this way. They actually have a signal that is a dual signal, so they're part of both this IPMS and this MAM ancestral locus. And so part of that is because the rate of evolution is slow enough that you can see signals in the Cleomacea of the shared relationship between IPMS and ancestral MAM. And so if we were looking at this in terms of phylogeny, so you have MAM over here in the Brassicaceae, and then MAM over here in the Cleomaceae, you can see that the Cleomaceae MAM is more closely related to all the Brassicaceae MAM, meaning this is a much more novel retention of MAM in the Brassicaceae. You can also see something odd in the Cleomaceae in that these Cleomaceae MAM genes have both this HMGL domain and a leucine dimer domain, which is not typical. That's not what we expect, and it's not what has been in the literature as far as the Brassicaceae. This analysis also showed us that indeed the beta duplication is the origin. So that first whole genome duplication event is what has allowed IPMS to duplicate, and specifically IPMS only wants to duplicate in the cases of whole genome duplication. That's our salt, I guess, in our cake recipe. And then this analysis has also given us insight into when the Brassicaceae lost this leucine dimer domain in the transition away from IPMS. And because of the duplication patterns, we can say that this loss occurred after the alpha event at the base of the Brassicaceae, which is important for understanding that. So a closer look at the Cleomaceae. So we see that the ancestral MAM experienced some amount of dosage constraint, although it is able to get up to 3 copy numbers. So when we're talking about numbers here, so these all, the ones with the triangle, so the ones with the leucine dimer domain, they share a dosage, right? A specific stoichiometry that they have to be at, or they might function improperly. So in this case, they seem to be ancestrally stuck at 4, so 4 triangles here. When we look at the extant Cleome violacea, we actually see some losses and novel gains, which is typical of what we see of MAM in the Brassicaceae, that at the MAM locus, you'll get these losses and gains of tandem duplication, but staying at that 4 number. And then in the Cleomaceae, there's actually a hexaploidy, or a tribal, a whole genome triplication event, which is not a single event, it's actually 2 events that happen in time. But we see that even through this whole genome duplication event, that 4 dosage is maintained. But then we look into Terranea, and we see actually there's 5 genes now. So we went from a dosage of 4 to a dosage of 5. And in looking closer, you see that the extra gene that is there has lost that leucine dimer domain. So because it has lost this leucine dimer domain, we now have a novel, and so this is actually a transposition. So there's both a novel context in the genome, a new address for MAM in the genome. It's also being expressed, and it's more expressed. It's more highly expressed in the leaf tissue than the other MAM genes in the Cleomaceae. Now, this is, since there's not a whole lot of biochemistry work that has been done in the Cleomaceae, we don't know if MAM behaves the same way in the Cleomaceae as it does in the Brassicaceae. So more work will have to be done both to confirm these patterns and to characterize that diversity. So MAM in the Brassicaceae. I was able to identify 7 lineages of MAM. So here is, this is a little complicated. So just know that each different color is a different flavor of MAM, and that this MAM ET here in orange represents that novel transposed gene group. And in the Brassicaceae, there are multiple lineages, but the ones included in my sampling are lineages 1, 2, and 3. And then I also included Aethionema. So in this analysis, I did a domain tree analysis and a full sequence tree analysis. And this is just important to point out that these are not congruent, which you would expect, since the domain is a conserved part of the overall gene. But since we actually see a signal of gene hybridization or gene chimerism, particularly in lineage 1, and including Arabidopsis. So these are interesting dynamics that complicate this story of this gene family. So to break down some of the transitions that occurred here. So I'm pointing out here that the loss of the leucine dimer domain occurred sometimes after alpha, and without that domain, MAM duplicates much more readily, and more MAM genes, you get more MAM action. So it's doing a lot more for these plants in terms of defense. So in this case, we get the origin of these B and D types, and particularly in lineages 3 and 1, you get a lot of tandem duplications that contribute to MAM diversity long term. Going up to the tribe Brachycea, which I'll talk a bit about more in a moment, what is novel are, so here we have whole genome triplication events that happen after actually the origin of that transposed locus as well. So you have now multiple addresses in the genome that give MAM room to diversify at specific loci and freeze one MAM type from the cis-regulation of another MAM type. So when I talked about the and or phenotypes, this is what I hypothesize is the reason we get an and type in Brassica species. So why Brassicas seem to be able to do both at the same time, while something in lineage 1, like Arabidopsis, is limited to only one majority glucosinolate type. Okay, so conclusions for this section. In the Cleomacea, the presence of the leucine dimer domain may constrict MAM to a shared IPMS dosage. And then in the Brassicaceae, we got 6 domain clades, and without the leucine dimer domain, the MAM locus readily duplicates in response to whole genome duplication. Yeah, and so I think the biggest takeaway for this is that it's not one gene duplication type that really plays into the diversity of this gene family. It's multiple happening at very specific times. If the alpha event happened, after that leucine dimer domain was lost, the entire landscape of the flavor profile of plants in the Brassicaceae could be completely different. And evolution would have happened very differently. So it's all a bit of a luck and chance game here. So for this next part of my presentation, I'm actually going to be talking about a project that has spanned a few graduate students, from Tatiana Arias to Jacob Washburn, myself, and then on to Sean Thomas. And so I'll be talking specifically about my little section of this, but understand it's a bigger project that is in scoping a lot of PhDs. The tribe Brachycea is one of 49 tribes in the mustard family. It's the most economically important tribe in the family. Again, people eat these species across the world. Just one, Brassica oleracea, comes in a myriad of types. But personally, some of my favorites in the tribes aren't the well-known ones. They're the weirdos. They're the Cacaili marittima that has this boat fruit shape that allows it to traverse the Atlantic Ocean and become an invasive species. There's the Crandy Cranberry hispanica that has been used as an oilseed, but has this spherical fruit shape and already comes with a radicle exposed within the seed casing. You have deserty types of plants to woody plants, and so the things that you would walk over and you would never recognize that they are actually a relative of your common kale. And then what's also really great about this tribe is the concentration of genomic resources, which allows me to do analyses that aren't necessarily capable in a lot of other systems. The ancestral distribution of the tribe is around the Mediterranean. Like Chris mentioned, myself and Mackenzie Mabry were supposed to be in the Mediterranean last March, right before the borders closed down. We're still hoping to get back and to do some collecting, and particularly because a lot of these plant species are under threat of sea level rise because they occur on beaches, and endemically on very specific islands. So there's a lot of places for us to potentially lose these species. Many of the tribe's largest genera are known to be polyphyletic. So what does that mean? That means the genus Brassica, for example. So ideally, you want all of the genus Brassica to share a common ancestor before anything else. That just is definitely not the case. And it is a well-known problem that this tribe is difficult to figure out evolutionarily. Part of that has been placed on the presence of this hexaploidy event in the tribe, and actually in a recent paper out by a collaborator, Yue Hao, they looked at the subgenomes, so the ancestral subgenomes that are involved in the whole genome triplication. So who were the parents of this polyploidy event? And you can see over time, you go through a process of fractionation or diploidization where you lose genes that you don't need extra of. So say we doubled our recipe, or in this case, we tripled our recipe. And then instead of just baking 3 cakes, maybe you went down a bit. So you remove some of the eggs and you remove some of the milk, and you got a different recipe from that because you didn't need as much over millions of years, okay, of cooking. All right. So just to break down some of the characters that we're talking, some of the clade characters that we're talking about. The green is representing this Rapa oleracea clade. Purple is going to be this Niger clade or black mustard. It's also home to Sinapis alba, the yellow mustard, and these are all determined by chloroplasts, chloroplast phylogeny. That's that boat-shaped Kakyle here in magenta. Cranberry represents blue. And so the chloroplast tree is like the mitochondria. It is inherited uniparentally. So you get it from your mom, not from both parents, but just from one parent. And so it's going to have a different gene tree, a different address and story to tell when we compare to nuclear phylogeny. So these are just, in this case, it's ITS, ribosomal DNA, but even in the case of other nuclear DNA, you see a similar pattern that comes across where their breakdown of these clades that are very strong in the chloroplast tree get to be pretty weak and confused and muddled when we get into the nuclear trees. So the goal of this study, watching the time here, was to test and adapt this genome-guided phylotranscriptomic method in a group that has experienced a recent whole genome duplication, and then, if possible, develop a resolved phylogeny of the tribe. So this method, like I mentioned, this is a legacy project. Jacob Washburn, he developed this method to look at, to scaffold transcriptomes, which generally don't have not generally, never have synteny information because they're more just floating sequence pieces based off of gene expression, but use genomes, an in-group genome and an out-group genome, to inform the synteny associated with those transcriptomes. And so what I'm adding is how can we use this in the context of clades with recent polyploidy that are a bit more problematic? What this does is, so in a normal case, you have like one gene across, and you'll take that one gene and it becomes an alignment, and you take that alignment and you make a tree with it. In this case, using synteny, we're able to take these paralog genes that each have their own address. So each different color would be a different address in the overall genome. And then you can make multiple alignments from that. So each one has an alignment, and then you can build trees off of those to inform your analysis. So this is the tree. There are a lot of takeaways from this, but I'm just going to focus on some of this core versus differences. And so one of the key things that we see is actually, so we still get that breakdown of the clades that we normally see in the nuclear phylogeny as compared to the chloroplast phylogeny. But there's a pattern where Cranby, this group here, falls outside of the core Corbiceraceae, which normally it falls within. And so you can see like a sliver of blue here in this nuclear tree, and even here. So you want to be paying attention to the nodes if you're not familiar with reading trees. Is sister to the actual nigra clade. And so this is just to remind you, oh, okay, this is what this weird Cranby shape is. And it's actually very distinct from the fruit types in the rest of this clade. So if you were a taxonomist, and I'm not saying I am, but if you were a taxonomist, this would be a much nicer tree to look at in terms of revising the genera, so renaming these groups, because Cranby is so distinct. If it fell out in the middle, you'd have to come up with 100 different names for, well, there are already 100 different names for these genera. So where did that pattern go, right? So actually, let me go back. So in this case, this is different from what we normally saw. And so I wanted to see why I wasn't getting what we normally see in the signal. Did I just do the analysis wrong? Did I mess it up? Luckily, because we have synteny information, I'm able to leverage the syntenic patterns. And so I actually took the gene list from this fractionation paper and utilized it in selecting genes, so I can look at, okay, what is the tree from this subgenome? And so these are only the single copy genes I took. And so these are the least fractionated genome, so the genome that is most intact. Then the most fractionated genome 1, so the one that is least intact, and then the one in the middle. And seeing if I break these up, do I get a different story than when I put them all together in the same context or in the same analysis? So in this case, when I look at the least fractionated tree, so we get this nigra clade sister to Cranby clade, and with the oleracea clade on the outside. So this is what we see in the chloroplast tree, and what has typically been seen. And so this is maybe what you would expect if you ran this analysis without the synteny method. And then looking at these most fractionated genomes, so these actually agree pretty much in terms of this this internal topology. In this case, Cranby falls outside, right? So that's this taxonomist likes this, that it's falling outside of this Corbiceraceae clade. And so we have two different topologies from these different sets of genes in the subgenomes of the distinct ancestors, the ancestral parents of these clades. So we have I also did a chloroplast tree and the nuclear tree here. So these are just simplified versions of these. And so when we look at the nuclear tree, we actually see that it matches these most fractionated, this most fractionated genomes, and the least fractionated genome is associated with the chloroplast tree. So in this case, we have two houses, each of these fractionated subgenomes giving us one topology, and we have another two houses, in this case, the chloroplast, and then the least fractionated genome giving us a completely different setup in our story. So what is the answer? Because both are right? There's one true history of how these things separated, but all these genes are telling the truth. It is like that riddle of, you find two people who are both telling the truth, but they have conflicting stories. And so I guess some concluding thoughts on this analysis. So I definitely was able to get a nuclear tree resolved from the core set of genes that show similar breaks near previous, and where Cranby falls outside of the core Brassicae. But the results also show the obvious hybrid origin, particularly of the core Brassicae. And so this is a kind of a rough breakdown of like what these topologies look like. So if you have a genome that looks like this, what is the true topology? Is there even a true tree for doing this? And part of this, I think, comes down to an actually age-old question for evolution, right? Are trees the best way of looking at and modeling species or lineage diversification? Particularly, like even Darwin, first wrote like, oh, maybe coral is a better way because corals can like web and reconnect and blah, blah, blah is better than a tree, but I don't think it's as poetic as to say the coral of life, as you would say the tree of life. And these results I think we're just butting up to it more and more as our technology becomes more powerful, and particularly in the case of sentient, we're able to see these patterns. And then I just wanted to also talk about this boat analogy. So if you watched the recent season of WandaVision, there is Vision has a conversation with himself regarding this idea of the ship of Theseus. And so the ship of Theseus is a philosophical tool to talk about when is something itself and when is it not itself. And so you have a ship that's sitting in a museum, and over time you can take wood from that ship and replace it. Maybe you're refurbishing it. And so you're constantly replacing it till maybe half of the ship's wood is not a part of the original ship. Now the display case for the ship still says ship of Theseus, but most of the wood was never a part of the true ship of Theseus. So at what point does identity come into play? At what point should we believe the lineage, the Cranby on the outside topology or Cranby on the inside topology, when both things are actually true? Okay, so I just wanted to take a quick shout out to some of my future directions. So I'm headed off to a postdoc, hopefully, and in August, where I'll be still working in the order Brassicales, but I'll be exploring some of the evolution of traits involved in photosynthesis and the evolutionary constraints of the development of C4 and other CCMs in the Brassicales. This is the hardest slide I've had to write, and if I missed you, I'm so sorry, and if I spelled your name wrong, well, I'm dyslexic, you guys know me. But I just want to thank my committee, my lab mates. They've been so much of help, particularly Mackenzie Mabry. We came in together in this program, and she's been my twin ever since. I don't know what my experience would be like without her. I have a robust academic community here. The wonderful, wonderful, wonderful Black women who who rode out the summer with me last summer during what was a very intense and emotional time. My good D&D friends here, people who have been a turn. I could not, being so far from family, I could not ask for a better community of people. I have in the Peers Lab, we talk about having a constellation of mentors and not just a single mentor. These are just some of the names of the people who have made me think about my science differently, both in the practice of my science, how to be a good scientist, a good citizen scientist, and how to do really good science. And of course, my friends and family and my cat, Jaja, who I'm glad is nowhere near here, so she can't bother me during this talk like she will on my other talks. And finally, I'd like to thank the enslaved Black people who helped to build this University of Missouri in Columbia. And I hope me going through this program is a part of the dreams that they had for themselves and the future for people. All right, thank you so much.
Fantastic talk, Sean. I was hoping I was going to have to throw a softball question at you while everybody put their questions into the Q&A versus the chat. So please put your questions in the Q&A. There's no time limit here, folks. We have lots of time for questions, but we can go a little bit longer as needed. Sean, before you start with the Q&A, which I know you can see, I just want to note that I love your 3 adaptations for this talk, updating our memes. The Syntony example, morphing from Ben Franklin to Black Panther is brilliant. The switch up of talking about dosage from our sushi analogy to cake. Cake is way better. Don't put in too much sugar or salt or baking soda. Who cares about the eggs and the milk and the sugar? And the ship of Theseus with the WandaVision update, that is brilliant and totally on brand for our lab. So good job representing. So with that, I think you can take on, it looks like there's already the first question from Jim Bergster.
Okay, so Jim asks, you mentioned that MAM has a dosage constraint when a dimer domain is present. What do you think the selection pressure is for this? Well, it forms a homodimer, and so I assume it can't function properly if it doesn't have that, doesn't have the right amount to pair with, so that stoichiometry effect. I think since IPMS is involved in leucine biosynthesis, I believe that if these MAM also have this dimer, and they're not, and maybe they're still able to pair with IPMS, and so then if they have it, then they're impacting leucine biosynthesis and messing up the whole primary metabolic process. But like I said, we don't actually know in the Cleomacea how MAM is acting, if it does interact with IPMS or it doesn't. But I assume something there is what's causing that issue. Great. Okay. Anon asks, I am familiar with the whole genome duplication in the fish lineages, but whole genome duplication appears to be rarer in animals than in plants. Maybe I hear more about in plants because of our lab, probably. But in this case, but is this really the case? I.e., does it happen more frequently in plants? So there are some great theories around why plants seem to have more whole genome duplications than animals, even though animals do have a lot. I think part of it is about an external fertilization, so you have more chances to get those errors in meiosis. So often fish and frogs, those are organisms that have polyploidy. There's also a sex chromosome hypothesis where if you have sex chromosomes, it's a lot harder to make it through polyploidy. So that could also be a part of that, why animals might have a less common chance of getting it. Joanna says, I have general knowledge question. What is it about plants and gene duplication is more common? Okay, so yeah, I think that's pretty much the same part of that aspect. Is it because we don't know other organisms frogs. Yeah, and there's some things that we thought were polyploid, but are actually just huge genomes. So it's also like one of those things that we're still figuring out, even for plants, that not everything that has a really big genome is polyploid, and sometimes there are other processes involved in that. Thank you for the question. Michael Vierling asks, what could be a risk if the dimers don't form due to unbalanced dosage? Yeah, so I think that's the interference in leucine biosynthesis is in affecting that primary metabolic process, which are critical to survival, right? Candy, thank you so much, I appreciate it. And then, okay. Paula McSteen asks, in your man tree, you showed that there were 2 in Cleomaceae, MX and MY, but it looked like MY shared a common ancestor with Brassica more recently than MX still had dimerization domain. Any thoughts on the difference between MX and MY? Yeah, I think it speaks to the Cleomaceae just having older lineages of MAM than the Brassicaceae. So I think all Brassicaceae MAM stems from a single MAM gene that lost that leucine dimer domain. I think once that loss occurred, it no longer needed those MAM that still had the leucine dimer domain. So it just stuck with that one. So it's like a bottleneck effect. So you just lost some of the older copies, where that same process hasn't happened in the Cleomaceae yet. Maybe it will happen in a few million years for Terranea that has that transposed loss of domain. Zach Miller, what are the physiological / reproductive costs and consequences of polyploidy in plants? For those that make it through successfully, are they associated with higher levels of species radiation across plant families? So this is another thing that's highly debated, because there are these huge species radiations that are associated with polyploidy and actually have like a lag time pattern to them. But polyploidy is very difficult. We have a lot of crops that are polyploid. And part of that is, we see a really big fruit. So polyploids often have bigger fruits and things like that. And humans want to take that and cultivate that. But in the wild, it can be very genomically shocking to be a polyploid and have to go through the process of polyploidy. So most polyploids don't survive, and just the lucky ones that do have access to a lot of extra genes that allow them to then innovate and their descendants to innovate in the future. Yeah.
Right. And I have a follow-up question. So in your first part on the Cleomaceae, can you say more about kind of the missing link analogy in terms of, like, was it really just that all the other people were confusing orthologs and alleles because they didn't use an outgroup? I mean, is it that simple, or is there more to like why you are now the world expert on this family of genes?
Well, I'm probably the world expert only because one of the real great benefits of working in the system is that there are so many people already working in it, and I just looked in a slightly different direction with a new tool and got to gain a bunch of novel insights. I do think species sampling is very important for these, which is hard because you're talking about genomes, right? And not, getting a genome isn't trivial, even though the technology is getting better. And so, yeah, I think the inclusion of the Cleomaceae did a lot for being able to interpret the data in the way that I have been. But even looking at my analysis, I wish I had more sampling in lineage 3 of the Brassicaceae. I wish, because there just seems to be so many more dynamics going on, but I just didn't have the power to say a lot of things with the level of sampling. Also, you want to look at these loci at a population level, right? Because this is an active population level dynamic where you'll have certain ratios of MAM in one population of Arabidopsis, for example, to another. So it's so many dynamics that I think it's pretty easy to believe that if you just miss something, you're going to miss a whole lot.
Well, I have another question then, since I have the floor. So you know that we have Alex Harkis led a big project to get a bunch more Brassicales genomes, and we have many more genomes coming for the Tribrissaceae, Brassica nigris, Cranby, things like this. What would be your dream, and maybe you're going to do this in your postdoc with Erica Edwards and Jen Weiscaver, of how you would use your tools to use all those new genomic resources to answer questions?
Yeah, well, so I am I'm hoping to learn new tools in my postdoc, of course, and to even develop some tools potentially to utilize some of these genomic resources. I'm really interested in systems biology broadly, so I think there will definitely be a place of leveraging all the resources. That's also a really great reason to work in this system, is that, you know, people just keep popping out genomes, so that, you know, I can continue to do things without having to go through the process of sequencing them myself. I just have to have good friends.
Looks like you have a few more questions in the Q&A now.
Okay, Thomas, oh wait, Kate, Michael asks, do you think that this method could be applied in the future to examine genes across something like 35 super pangenomes? Oh, pangenomes. Yeah, I don't know. Pangenome, it just adds a whole extra layer of computation that you'd have to include to look at pan. I think that's probably where we're headed, but I'm not sure if the algorithms or the analyses have been written yet to include that. You can't just throw a pangenome into the pipeline, but maybe if you separated it into a bunch of pangenomes, I don't know. The computational power sounds pretty intense there. Thank you, Michael. Thomas Rice asks, what is your D&D class and race? I'm partial to druids. I'm, very nature themed in my life. I'm wearing floral, so I think that's generally there. And and I like a good, right now I like a good warforged, something about robots or whatever. Yes. Yeah, thank you all for being here and experiencing my talk.